My name is Mohit Mazumder, and I am a bioinformatician by education. And since last 12 years, I've been practicing and applying bioinformatics for research, and now both research and for collaborations and for education. So during my PhD, I was working on several drug targets and key proteins, as I was associated with a structural biology lab, and that had led to several opportunities to actually work on new structures which are not even published, and then what are the different ways to analyze those structures, what are this, how much they are talking about the function, and what are the different ways we can correlate structure with function, and then probably design several strategies around it. So as I was telling you that in my PhD, I was working with several key drug targets, and one of the objective was to understand the site-specific mechanism. So sometimes the objective of doing a drug discovery project or structural biology project or understanding the cheminformatics that would be required to give us the desired result, it is important to understand the specifics of the very much detailed version of what you can look at and what you can find in literature in different databases, and then develop a model. So that would be the purpose of this entire program. So understanding the site specific differences, mechanisms, comparisons, and I'm talking about three-dimensional structures here, and then developing strategies to overcome challenges specific to structure based drug designing, and learning about how what would be the next steps. So what would be the next steps for after the structure based drug designing, and understanding where molecular dynamic simulations and quantum mechanics, all of that are applicable, and where are the innovations that are happening in the industry with AI and the deep learning. But to give you a perspective, this is a chem informatics and biomedical drug discovery program, and we should be able to together visualize how all of this would be, because all of this will be a visual experience. So you'll be working on three-dimensional structures, and at the same time, you'll be working and looking at small molecules. So let's start with this slide, which which shows the emerging drugs. So to understand that, I mean, how the drugs actually look like, it's a slide basically describing that the FDA's Center for Drug Evaluation and Research, which has approved 16 new medicines for now, and 15 noble drugs were approved in 2021, and 53 novel drugs were approved in 2020. So in 2022, the potential new drug targets are, you can see that there are big companies involved in this. You can see Novartis, AstraZeneca, and then different types of molecules which are like more target based. So if you can see that AstraZeneca's search for treatment of ER positive breast cancer, which is more specific to individual mutations, are also also described in this slide. So talking about malaria, new drugs on malaria, or the drug that Biogen came up with for autoimmune diseases, and then for Bristol Myers, the ubiquitinase mechanism targeting that, and ligase modulation. So these are target-driven drugs, and this is described with a mechanism of action, and we can perhaps maybe understand some of these case studies in the program going forward, that how drug repurposing is also one of the ways we can counter, or we can find solutions to some of these diseases. And today in this session, we will be using one of such case studies, which is a straightforward case that we will be looking at. So thank you for all of you for starting to share about your background. It's really helpful to learn, starting from Kritika, I see Herschel, I see Cynthia, I see Harish, Sashwati, and then Sana and Abigail and Broana and Arpan and Michaela and all of you. So it's thank you so much for sharing about your educational background and where you are from. I see that we have a lot of students from or participants from Nigeria, and we have a lot of also ongoing programs going in Nigeria and Africa. So good to see participants from South Africa and India. And I'm hoping that we will be able to help you with some of the structural biology related queries, structural biology related ideas, because in this program, the one of the objective is to develop that idea into a project, and that's why Sonalika specifically discussed about that in the beginning and shared also the project submission form. So please feel free to write about your ideas. Maybe you don't have all the components there, but then you can obviously connect with us on Skype and have those discussions, and we will be able to answer them not right away, but whenever we will be able to see them, we will be able to communicate in that sense. So when you're communicating on Skype and when you're communicating in other places, please be patient as we are working all the time, but we'll be looking at those questions and we'll be helping you out with answers. And then we have these sessions planned as well. Along with that, there are recorded sessions and videos and all of this. So I think we will be able to cover all of that. Okay, hello, Metter. So, Andrea and Clement and Ishaku. So, thank you again. And now about the context of the program. Now we are moving towards the actual session. So, in the context of the program, this is the Learn program page, and we will be running this program for at least 3 months, which is also one of the subscription levels. So to those who are subscribing for 3 months or 60 days, 2 months or 1.5 months, 45 days, we will be able to cover those objectives within the time frame. Now about today's session. So today's session is focused on some of the basic concepts that are really important to start from the beginning and develop an understanding and holistic understanding of how structural biology or computational structure biology is being utilized by researchers and utilized by industry. So understanding the molecular structure of biological macromolecules, because those are the target structures, then understanding what are the technologies that are being used right now to generate such information, and that technology could be about the way we have evolved using X-ray crystallography, now cryo EM and all of that, and then what are the way to look at this. What are the primary, secondary, tertiary, quaternary, and structural domains, structural motifs, protein subunits? So many of them are covered in basic textbooks, and I guess many of you are kind of aware about all of this, and perhaps anyone can Google some of these keywords and get a better understanding from the search results by going through Wikipedia and all. So our focus will not be, to go through these topics just like a traditional way, but what we will be doing is like we will be working on some of these topics, do some hands-on and understand from a case study level. But again, we for those who are from not from a structural biology background or have never heard of some of these keywords, we have put together these lessons that will be helpful for those who wants to understand. For example, what is structural biology? Structural biology is the study of how biological molecules are built using a variety of imaging techniques. Scientists view molecules in 3 dimensions to see how they are assembled, how they function, how they interact. So that has helped researchers understand how the thousands of different molecules in each of our cells work together to keep us healthy. So structural studies have shown how mutations or mishaps molecule make us sick, and as a result, studies have prompted in new treatments for many diseases. So that is one of the objectives. What are molecules? So molecules can be grouped into two or three atoms held together by a chemical bond. So molecules, we will be discussing about RNA, we will be discuss about DNA, proteins, carbohydrates, sugar, and lipids, fat. So structural biologist, as a structural biologist, these are very particularly interest gives a lot of interest because of the nature of this to be protein, and then we are investigating these large molecules made up of combination of RNA and protein, and then RNA protein complexes. So these proteins are molecules that contribute virtually to every activity in the body. They form hair, they form fingernails, they carry oxygen in the blood, allow muscles to grow and move, and much more. So proteins are made of long strands of small molecules called amino acids, and we will be looking at amino acids, how they look in three dimension, and what are the way they move, and what are the different side chains and all of that. So there are 20 amino acids found in nature. Each protein contains a unique combination of few dozens of many thousands of amino acids. So some proteins consist of multiple amino acid strands that are wound together. So one of the important aspects of discussing about all of this is also like the shape. When we are discussing about the shape, we are discussing about the three-dimensional or the tertiary structure, and we are talking about the strand, twist, bends, and folds. So then we are discussing about how to understand the way protein folds. So we, in this context, would also be discussing about the new AI-driven, AI-guided DeepMind AlphaFold, and we'll discuss that how protein folding has been almost now much more easier than to do this before. So all of this, I mean, now anyone can do this, and students from high school, students without a background of biology, and specifically computational biology, will be able to learn and go through this training and learn how to apply this and how to use this. So we have a couple of lessons that describes the computational structural biology that we will be covering in this session, and the next session, introduction to cheminformatics, where we will be discussing about the chemical world. So the different aspects of proteins, like we will be discussing about these different case studies, which are about different proteins. So different proteins have different functions, and their shapes or specific functions help cell hold their shapes and move and movement and all of that. There is a type of protein called transporter proteins that are embedded in the cell's outer membrane and form channels that helps in vital movement of vital substances such as sodium, calcium, that pass into or out of the cell. So, I mean, in the context of drug discovery or to develop new drugs, so drug typically work by either blocking or supporting the activity of specific proteins in the body. So we will be discussing an approach called structure-based drug designing, where scientists usually make a template for a protein, use that blueprint to create new medicines. So that's the mechanism in a layman's word. But to do this computationally, we will be able to do this in an in silico environment, where the thought process will be to mimic what is happening within the body or within the system. So computer model would allows us like to examine this in several ways, and that's why we were talking about molecular dynamic simulations, or we are talking about molecular docking simulations. So I mean, there are several examples now where people have determined this or have gone into different clinical stages and also come up with a drug structure-based drug, and now knowledge-based as well. So there is a lot of information and there is so much databases. And now what we've been able to do is connect that information, fill up those, connect those dots, fill up those blanks, and that utilizes, deep learning, a lot of deep learning, and a lot of structure-based or knowledge-based drug discovery. So all of this could be learned through the coursework that we've been discussing. This is a short session, so one of the objective would be to for you would be to understand this in the context that where are these resources and what we can do when before we meet again. So these are some of the things that you can do on your own, and together with the help of mentors, we will be able to work on some of these aspects. Now, the first thing we need to do to start to get going is to understand that where is this data is available, and how to interpret this data. So today we will be doing that. So protein data bank archive information about 3D structures of proteins, nucleic acids, and protein data bank is a free repository where we will be going and doing some of our analysis, understanding where to find this information, how to interpret this information. So I was talking about the three-dimensional shapes, but in terms of proteins, you can see here in this image different types of proteins, and like really all kind of weird shapes, and like really, I mean that kind of explains the diversity in this area. So now what we would want to know about this, how complex assemblies and this understanding helps students and researchers understand all aspects of biomedicine. And we will know about application in biomedicine, which is quite evident, but it has application in agriculture, from protein synthesis to health and diseases. So the RCSB PDB builds upon the data by creating tools and resources for research and education, as well as education in molecular biology, structural biology, computational biology, and beyond. And one of the things that we will be discussing in this context is about a case study that would be on SARS-CoV-2, uh, COVID-19. So in 1.5 year of the World Health Organization's declaration, within 1.5 year, when WHO declared that COVID-19 is a pandemic, over 1,000 structures of SARS-CoV-2 biomolecules have been deposited in the protein data bank. It's a rare to obtain such a it's very rare to obtain such a snapshot of intense effort in structural biology, and that happened, and still there are so many structures coming up, especially with cryo EM now, and then there are other repositories. So here in the image you're seeing protein data bank, you're seeing other repositories which are maintained by EMBL EBI. So um, in that context, it's important to understand where to find the information, where to find these sort of information, which is already available, which can actually help you save a lot of days in your research. It will actually help you explain this to someone else in a very simplified way, so that you can go to the next level. So that's why that's the objective of the session. And Uniprot, for example, if you just search in Uniprot, is one of the world's leading high quality, comprehensive and freely accessible resource of protein sequences and functional information. So similarly, after these are the databases and repositories which are maintained by EMBL EBI. So we will be looking at the repositories that are maintained by NCBI in the NCBI portal as well. But in today's session, we will be looking at some of these repositories. So about PDB E is again the founding member of the worldwide WW Protein Data Bank, WWPDB, which collects and organizes and disseminates this data for biological micromolecular structures. So there's a lot of information, structures that could be downloaded from here, and understand the chemistry, some of the chemistry, how it is connected with it. So EMBL has also developed and integrated with AlphaFold. So AlphaFold is a AI system developed by DeepMind that predicts a protein's 3D structure from its amino acid sequence. It regularly achieves accuracy competitive with the experimental structure. So I have been, talking about this AlphaFold, and in many of my webinars, we have gone through the way AlphaFold came up, and also we have gone through one of the video which explains AlphaFold. So just let me ask you this question, like how many of you know about AlphaFold, and are you interested to look at the video of how this came up, or we would you want to skip that part? So can you tell me in the chat, please? So do you know about how much you know about AlphaFold? You're interested in the video, or let's skip that? So as I was telling you that AlphaFold is an AI system developed by DeepMind, and it predicts protein structures, 3D structures from sequences. So DeepMind and EMBL, European Bioinformatics Institute (EBI) has partnered to create an AlphaFold database. So the AlphaFold database, which is what I'm showing you on the slide, is to make these predictions freely available to the scientific community. So, and since it has been an year that in the CASP 14 they did this, and then it has been a year. So since then they have done they have done 200 million predictions. So they have done 200 million predictions, which is available on this website. That is what I'm showing you on the screen, and we will be sharing the links uh, over the email. I think Sonalika does told you about that. But after the session, there will be an email that will have all this information that we are covering, and then you will also get the mm, video recording. So that's another way. So right. So as I was saying that the latest this database contains 200 million protein structures and provide a broad coverage of UniProt, where you will be able to see protein sequences as well as the annotation. So it's a great place to do what? It's a great place to come up with research proposals and problems and finding those proposals and problems. So you can individually download that in the context of human proteome and 47 other key organisms. So you can target organism wise, look at the structures, predicted structures, and then hypothesize from there. So it's very becoming much and much easier. It's just that one has to look at it and then start digging and then develop a proposal. That's what I will be expecting from some of you before we meet again. So please, go through these resources and then look at some of the interested protein structures, interested proteins, or interested organisms that you would want to go further, investigate further, and then we would be able to we would be able to do this. So let's move to the next video. I think you said that you are interested in the video. So in the next session, when we have the AlphaFold also planned, so these are some of the links, GitHub links, Google Colab notebooks that we will be going through. So today's session, we are not doing Google Colab, but we will be having an basic introduction to Google Colab, and then we will run some of the Colab notebooks and run some predictions using Python, Google Colab.